Welcome, and thank you for joining us. My name is Dean Lee, and I'm Professor of Physics at the Facility for Rare Isotope Beams, or FRIB. Today's public talk is part of an initiative at FRIB called the Advanced Studies Gateway. The goal of the Advanced Studies Gateway is to inspire people. We bring together researchers, innovators, creative thinkers, artists, and performers from all fields and strengthen ties between Michigan State University and the community. Today we have the great pleasure of presenting a public lecture by Professor Tatyana Sharpee from the Salk Institute of Biological Studies, entitled How Hyperbolic Organization Facilitates Learning in Biology. Professor Sharpee received her PhD in condensed matter physics from Michigan State University, studying under the supervision of Professor Mark Dickman. After her PhD, she started to work in computational neuroscience at UC San Francisco, where she developed statistical models for analyzing neural responses to natural stimuli, which exhibit strong correlations and non-Gaussian effects. These methods made it possible to reveal new adaptation processes in the brain by comparing neural responses to white noise in natural stimuli. Her independent research program has started at the Salk Institute for Biological Studies, where she is currently a professor in the Computational Neurobiology and Integrative Biology laboratories. Dr. Sharpee is a fellow of the American Physical Society. If you have any questions during this talk, please type them into the chat or the question and answer dialog box. At this time, it is my great pleasure to introduce Professor Sharpee.
Thank you very much for the introduction and for the invitation. It's a great honor for me to participate in this series, not only because of other people who have preceded me in this series, but also because invitation comes from Michigan State University, where I received my PhD. So I think it's an extra pressure for me to report on what I have been doing for these past 20 plus years, and in a way, perhaps it's my another thesis defense today. So I would like to share with you my passion for hyperbolic geometry. I will explain you what it is, and I will try to convey why it is important and why I think it not only describes the structure of our universe on cosmological scales, but it also permeates all levels of biological organization, from cells to neurons to the brain to our perception, and why I think it's interesting. So the work that I'm presenting today is the result of primarily 3 PhD theses by Yuancheng Zhou, who graduated in 2021, Huanqiu Zhang with a PhD in 2023, and Anup Raturu, who will be graduating in a few months. So let me begin with an introduction to geometry, and most of you, I think, heard about Euclidean geometry in your high school classes, and it begins with Euclid. This is his painting about him, and he famously summarized the mathematical knowledge prior to him in his work about the elements, and postulated, based on 5 postulates, he created the so-called Euclidean geometry, the geometry that we use in everyday life. And one of the postulates is that is through a parallel line, through one point, you can only put one parallel line. Another person that I wanted to mention is René Descartes, about a few centuries later. So the joke says, to introduce the Euclidean geometry, he was sick and he was lying in bed and he was watching a fly right here, pointed here. And as the fly was crawling around the ceiling, he thought that, oh, I can use the coordinates along the 2 edges of the ceiling to describe position on the fly of the fly, and that's how we now use Cartesian coordinates. So use your downtime wisely. You can come up with the new geometrical constructions that but it basically evaluates that the distances are, in Euclidean geometry are based on the square of the distances along x and y coordinates. And we think in our everyday life that, that's cosmological scales aside, that's what describes our geometry. But actually, it's actually not true. So one can do quantitative measurements of how we perceive space around us, and if René Descartes was more quantitative and could measure his distance perceptions in a more fine detail, he would recognize that his perception of space is actually hyperbolic. And we'll talk about this, but for example, you might, there's this common notion that a baby is trying to grab the moon. So it turns out that our perception of how the distance to the moon is actually much closer than its physical distance. And that relates to distortions that occur in our perception when it comes to distances. And I will describe the advantages of these distortions. So based on Euclidean and non-Euclidean geometry, I would like to give you some history. So from going to Lobachevsky, so we are from Descartes, we will go to Lobachevsky. He was wondering about these 5 postulates of Euclid, and one of them, the 5th one about the parallel lines, it turns out that if you remove it, there is no contradiction. And he tried to publish this, his paper on hyperbolic geometry. It was rejected from when he submitted to St. Petersburg Academy of Sciences, and he was published in a not very well read from his university in the middle of Russia, Kazan University, in the proceedings of that university. And in parallel, Janas Balai also published a few years back in an appendix by his father, in a textbook by his father, also the same ideas about hyperbolic geometry. And if you are interested in art, then I'm providing here a painting by Escher, a Dutch painter who popularized hyperbolic geometry in many of his paintings. He has a series of 4 paintings, and this is one of them, about demonstrating how you can fit the whole Euclidean plane or Cartesian plane, as in the René Descartes version, and squeeze it into a circle, and infinity would be the boundary of this circle right here, unattainable. So all of these fish that you see in this painting are all of the same size, but as you approach the boundary of the space, the fish get smaller and smaller, but that's the kind of epitomizes the horizon that we would like to approach. So I will be describing to you the peculiarities of this geometry and how it is related to many levels of biological organization. So we became interested in the hyperbolic geometry for about 10 years now, and we find it in all sorts of circumstances. So I will first summarize the situations where it arises, and then describe why I think it's general. So first, for example, in natural order statistics, the statistics of order, and so famously, if in color perception, I can specify color by 3 coordinates, and if you would like to match the color of, you need to paint your walls, you can go to a store and say, well, I need color with these coordinates. and they will be exactly able to match the color. But the question is for odorants, for example, we do not have a quantitative map for odorants, and that's what we were trying to do partly in that paper on natural odor statistics, to develop a hyperbolic map for odorants, such that next time you order wine, you can say that your preferred coordinates for wine are 5.3, 2.5, and 0.6. So in mammalian gene expression, so it means which genes are expressed in all cells, in which cells and in how. So that turns out also to be described by hyperbolic geometry. In neural representations, that's what I will be focusing on today, in the brain of how we represent space, and also in viral evolution. So why do I think that hyperbolic geometry is important? So there are two reasons, and they are related to each other. One is very practical reason. It turns out that, and this is what I alluded to with this Escher's painting, is that it describes hierarchical tree-like networks. So in many types of situations, it can be in language models, and in other circumstances, the data is described by nodes that are increasingly more and more refined. We can think about concepts of how a child learns language. First, it can be an animal, and then it can be a cat and a dog, and then what kind of cat and what kind of dog. So then it cascades down. And the same thing in, for example, in cells. Cells will come in different types, and then they will become increasingly specialized, whether it is a skin cell or a muscle cell or a hair cell. And also in machine learning right now, as you know, there is a lot of interest in these hierarchical models, and in machine learning and deep learning research, they also like this hierarchical hyperbolic representations. So one peculiarity of hierarchical organization, that if I want to compute distances between two nodes, I do not compute it in a straight line. I have to go back to the last common node and then come back out to the edge of the network. So as a result, the trajectory of the shortest distance between points is not straight, but curves, and it curves to the center of the network. If you imagine creating a map of the space with this discretization, then the corresponding continuous approximation would also be discrete. And there are two visualizations of the hyperbolic space due to a French mathematician, Henri Poincaré, who was very much interested in this geometry. And one of the visualizations is called Poincaré half plane, and you can think of this as discretization of the space that goes along with the hierarchical network. So approximately in the Poincaré half plane, the trajectory that describes the shortest distance between points will be the semicircle that goes between points A and B and goes deeper into the center of the space. And the curvature of the space describes how fast this space branches, or how deep is the network. So another visualization, is also Poincaré, and in this case, a Poincaré circle, where the number of points is uniform along the, if you imagine sampling the tree, then you will find out that most of the nodes will be at the edge of the tree, and the shortest distances will again be curving towards the center of the space. And this is opposite of the life, for example, on a sphere, where if I wanted to compute distances between 2 points on the edge of the sphere, I will go in the opposite direction on the outer side of the boundary. So that's one of the features. So why is the practical reason is that because in many types of situations we are dealing with hierarchical data, therefore, if our geometry is more closely approximating the data structure, it will become more accurate. And that's what people find in machine learning, that when you use hyperbolic geometry to create maps of data, they are more accurate. So that is a practical reason, meaning if data is hierarchical, you use hyperbolic geometry. And but then there is a second, more fundamental reason, and that is, why is data hierarchical? And here there is a fascinating series of papers by Dmitry Kryukov right here and his collaborators. And I will just quote here one paper from about 10 years ago. The title is Sustaining the Internet with Hyperbolic Mapping. So they were interested in understanding, or they were solving problem that as you add nodes to the network and the network gets bigger and bigger, how are you going to communicate between different nodes effectively? And in general, it's a very difficult problem, because in order to find efficient paths from one node to another, each node should have a complete knowledge about full connectivity in the network. And it's impractical because nodes go up and down. And me as a computer node, I don't know what is the status of the rest of the nodes of the network. And what they're interested in is how to develop effective algorithm for navigating in the network and communicating between different parts of the organization or the network using only local rules. So I know my coordinates, my status as a node, and I know where I need to send information. So they discovered that if we assign, or embed in the scientific terms, if we assign coordinates to nodes according to a hyperbolic plane, then they could communicate effectively between different nodes by sending, using only local information. So I know my coordinates, my hyperbolic coordinates. I know hyperbolic coordinates of my target node, and I know hyperbolic coordinates of my neighbors in the hyperbolic plane. Not necessarily in the physical sense, but in hyperbolic sense. Then I'm just sending my information to the my nearest neighbor that is closest along the eventual path towards the target node. And they found that using this local information, one can effectively communicate with the network. So in other words, that so meaning that I send information towards the node, it will be a hub node that is closer to the center of the map. Notice that all of the trajectories in the hyperbolic space, they always go towards the center and then they come out. So the fastest, which I think agrees with everyday's intuition, is that in order to communicate in the network, I would like to send information to a hub node or somebody closer to the center of the network and then back out. And how closer to the center of the network I need to go depends on how far is the target node. So if the target node is nearby, you don't need to go all the way to the center of the node. You can just come a little bit. But if the target node is beyond certain critical angle, then effectively you will be going back to the center and back out. So they found that using this hyperbolic organization, they could effectively communicate in large networks. And of course, this problem is very acute in biology, for example, or even in neural circuits. For example, neurons can die, or if they don't die completely, they can get tired. The connections can be strengthened or weakened. So especially in biology, the communications in the case where the structure of the network is plastic is very important. And so I think that these two reasons that I'm citing here are related, meaning that in order to achieve effective communication, it's good to organize network according to a hidden hyperbolic structure. as a result, the data is in a hierarchical manner. And because of this, reason number comes about the reason number one, where the hierarchical, most of the data in biology has a hierarchical organization. And then there's a third reason, which I think relates to hyperbolic organization on the cosmological timescales, is that on the cosmological timescale, the speed of light is finite, but in our case, the speed of propagation in the network is much slower. So we see these hyperbolic effects sometimes on the scales of meters in the case of human perception, because it takes finite time for example, for me to hear the words and then to perceive them. So there is a finite response time for signals that propagates within our brain. And so that finite response time determines the range of hyperbolic effects that we see in perception. So today I was going to focus on responses in the brain, but I'm happy if people have questions to go over the other examples. And I will tell you how we'll represent space. So for this Zoom lecture, we didn't have to travel anywhere, but often when I go to conferences, I say, how do we represent space? Is that, well, I don't need to know exactly how I'm going to get from San Diego to Michigan State. All I need to know is how to get from San Diego to San Diego Airport, from my home to San Diego Airport. Then there is something happens in the air, and then I need to figure out how to get from the airport to the department. So you can see how the representation is compressed. And the same thing is true for how neurons represent space. So a few years back, a Nobel Prize was awarded for figuring out how neurons represent space in the brain. So there is a region of our brain called hippocampus. And inside this region, there are neurons, many millions of neurons. And each neuron is responsible, its job is to send signals, which is neurons respond with 0, 1 signals in binary matter. So imagine this is a depiction of an experiment. This is a theorist depiction of a mouse. And as a mouse is running around, every time in a certain environment, every time the mouse passes through a certain region of space, that neuron will get light up. So you can see how in the brain the space is partitioned into these response regions. So one neuron will have a response region here, another neuron will have a response region somewhere else, and these response regions are known as place cells, and they come in all kinds of shapes and sizes. So that's kind of is depicted in my graph on the right, where you will have some neurons with very small place fields, which convey detailed information, and some neurons with larger place fields, which give you a bigger representations on a bigger scale. So numerically, this looks like this. So you monitor how the animal is running through the space, and you observe activity of many neurons, and each neuron is on the y-axis here, and the time is on the x-axis, and you see that some neurons go up and down as the animal is progressing through the environment. And when these when neurons go up and down together, that's perhaps because their place fields overlap, or because their place fields are more related. So there's a large body of work to understand how the animal can interpret these signals and figure out where it is located. So that in that line of work, we also became interested in analyzing the structure of these representations, and in particular, whether any systematic changes in the distribution of these response regions as the animal was exploring the environment and learning about it. So here I would like to do another historical note, this time going back to Darwin, and it's the everyday illustration about the use of mathematics. So it turns out that Darwin knew of this trick that we are about to use. And it's very natural for place cells. So imagine these are the place cells that we recorded, some small, some large, some overlapping. And what Darwin knew is that if you lift this representation into another axis, another z-axis here, so the position along x and y plane will be the center of the actual response region that was recorded, and the size of the circle will be the z coordinate. So automatically, just by this construction, you will be able to create a hierarchical representation of the data. It's the same data. I can use data as overlapping circles, or from this, I can create a tree. So the rumor has it, well, the published rumor in the paper. So turns out that before Darwin, species were described as a set of overlapping circles. So this is a picture of species, and here in the center you can do Homo sapiens, so the human, and people were playing games in the parallels of Europe, saying how can you classify species based on their flowers, based on how many petals you have, or other external characteristics. And apparently you can classify them very well by these overlapping circles, but just transforming the data into a tree can lead to a new level of understanding, and that's apparently how Darwin came out with History of Life. So our goal is to use the same method, essentially, is we have these overlapping circles in space, and we would like to put them into a tree to find out which neuron is the master neuron. It determines whether I'm in this environment or not, and which neurons provide a more detailed representation, and whether there are any advantages to using this hierarchical representation as opposed to covering the space uniformly with the same set of circles. So it's a collaboration. I collaborated with Professor Albert Lee, who was at that time at Genelia Research Campus near Washington, D.C. And they were interested in representation of very large environments. So it was a linear track, and it was 50 meters long. And even in a large institution like Genelia Farms, they had to stuff that 50-meter track into one room. And we will see that we will actually be using this curviness of the tract as one of the features in the data set. But that will be a little bit later. For now, what they were recording is this size of these response regions as a function of the position of the linear track. And I'm showing you the snapshot of the whole data set, and you can see that some response regions are very small, and some of them are very large, so can span a few meters. So this is data from a rat. And as a first test of this hyperbolic slash hierarchical representation, you can plot the distribution of cells as a function of their size. And if it is indeed hierarchical, or then you would expect an exponential distribution of field sizes. And that's what we observe. So one can use it as preliminary evidence that our representation of space, by our I mean in a rat, but hopefully generalizes to humans, is hyperbolic. But more quantitatively, so we would like to, as often, this is the idea, but the practice is a little more nuanced, because neurons can have multiple place response regions. Turns out that we go to a new environment, the same cell can have a different place field, so the cells are reused. And also, as you unroll and make the space bigger and bigger, the same cell will start acquiring multiple response regions. So what I have shown you with this hierarchical construction is actually more for intuition, but quantitatively we have to measure the distances between neurons based on how often they go up and down together. So once you get these distances, you can start to figure out on which surface these distances fall. And turns out that if we talk about surfaces of constant curvature, there are only 3 options, which is very nice. You can live on a sphere, and the sphere can have different radius. That's positive curvature or spherical geometry. You can live on a hyperboloid, which is negative curvature, and it can be more curved or less curved. Or you can have zero curvature, which is a standard Euclidean geometry. And once we measure distances between cells, these are like constraints. And using these constraints, you can try to put data on different surfaces like you would do on a mannequin. So for example, if I give you distances between major cities on Earth, then these distances will not align with a flat surface. And if I take local distances within maybe one country, then it probably will be an okay approximation. But if we start taking distances between New York and Sydney or across continents, then the distances would not add up. So by taking the distances between neurons, we then tried on, like on mannequins, on different surfaces of different curvature and of different dimensions, and see which surfaces are potentially consistent with the data. So in practice, it looks like this. So it's a little technical aside, is that you measure distances between points, in this case neurons, in the data, and you also place distances on different surfaces randomly, and you measure distances between what you would expect for the same number of points put on another surface. And then you can have multiple choices for the method with which you can evaluate the distance matrix from the data and the distance matrix from the mannequin, from the model. In this particular case, we used the topological method. And advantage of this, if any questions, I will go over through the details, but because because it's a public talk, I was just gonna say you derive signatures of different matrices, and their topological characteristics known as the number of cycles, and they will behave differently if the distance is generated at random or if they come from some geometry. So for example, this is Euclidean geometry, and then this is in random networks. So for example, you see that the peaks are increasing with the order of these cycles. So one cycle can be whether it is similar to a circle, the other one whether it's similar to a pyramid or in higher dimensions, or in the geometric model they will decrease. And then if it is Euclidean or hyperbolic, the rate of decrease in the position of these curves will be in different places. So one can use this method to figure out whether the data points that were collected are consistent with hyperbolic or Euclidean geometry. And so when we did that, we figured out that the data curves, which is in dashed line here, are consistent with the three-dimensional hyperbolic geometry because the predictions of that is the solid line plus the spread. So in other words, with the 3D hyperbolic geometry, it means that the rat has roughly two-dimensional representation of space plus one hierarchical dimension, gives you 3 dimensions in hyperbolic space. And the data, which is the same curve here, a dashed line on the left and on the right, is a dashed line is clearly not consistent with the 3-dimensional Euclidean geometry. And you can take data from different groups, and also not from linear environment, but from a square environment, which is much smaller. In this case, so linear track was, say, 50 meters, and the square data was on the scale of 1 or 2 meters. And even though it is much smaller size of the data of the space, you still see that there is a hierarchical hyperbolic organization that accounts for the data. So again, the data is in the dashed line, and predictions of different theories, either hyperbolic on the left or Euclidean on the right, are either consistent or not consistent with the data. So now I would like to tell you 2 interesting facts about this representation beyond its hyperbolic character, and something that I alluded to in the title, for example, that how it facilitates learning and what happens when you explore the environment. And it turns out when Huanqiu Zhang, who was the student who was analyzing this data set, analyzed data from different exposures. So the animal is brought into this environment, and then a day passes or another day passes, and they're repeatedly brought into the same environment multiple times. Then the radius of this hyperbolic map that we reconstruct from the data gets slightly larger and increases in time. So there is a joke that whenever a theorist is giving a talk, there will be a logarithm. So here is my logarithm. So the data is increasing as a logarithm of the amount of time that the animal has spent in the environment. And it turns out that this logarithmic increase is actually what you would expect if you measure the maximal amount of information that you can learn in discrete episodes of some time delta t as a function of time. So this is also, not just the curve is a fit, but it also has another explanation that this is the amount of information that could possibly be transferred in the amount of time t with a coefficient that measures the time scale of learning. And now it turns out that the size of this representation actually is optimized for the available number of neurons. And let me show you, give you the intuition for why that might be. So we did simulations with, you place these response regions as in the experimental data, and as a function of how many response regions you have. So suppose I only have 50 neurons and I have to represent certain size. If I build a very hierarchical and detailed representations and I only have 50 nodes, then most of the nodes will be empty. So on this side of the curve, when I build a very detailed, large hyperbolic representation, but I only have 50 nodes, then my representation of space will not be efficient. On the other hand, if I build a very flat representation and with no hierarchical separation, then I'm not taking advantage of the best way to represent space. So turns out that for a given number of neurons or response region, there is an optimal degree of hierarchical representation. And the more neurons you have, or the more response nodes, the larger is the optimal map, so the size of the map. So one could see that if it's better to use, in other simulations that I'm not showing, it's better to use hierarchical representation than a flat Euclidean representation, which will be kind of on this side of the graph. But for a given number of neurons, there is an optimal size of the network that you should build, such that you can fill it. And if you take this optimal radius right here and plot it as a number of neurons that are simulated, it falls on a straight logarithmic curve. And the interesting part is that the curvature that we estimate from the data matches the estimated number of neurons in that area of the brain. So in other words, the curvature of representations that is observed, we only record from a small segment of the brain, but the animal presumably knows what its capabilities are, and the curvature of that representation is a match for what you think is the optimal for the number of neurons available for the task. So I would like to summarize this part of the talk before I give you examples from brief examples from other levels of organization. So what we find in the case of the hippocampus and spatial representation, that with time, representations become more detailed. So you add more localized response regions, and the network grows, and the size of the network, or its radius, increases logarithmically with time. And its size is optimal for the available number of neurons. In other data that I haven't shown that I alluded to is that actually as the animal is running along this curved path of the track, if it slows down, it locally spends more time in that environment. And so for that part of the space, the representations become more detailed. So the primary determinant for how fast the network will grow is time. And in other words, when you're learning something, we often are told, take your time, because you need to take your time to allow for the neural connections in the brain to form in order to form the corresponding detailed representations. So now I will give you a brief example in viral evolution, just to demonstrate a generality of these findings. And so we were fitting the same methods can be used in a different domains, for example, in the genetic sequences of the virus, and evaluating distances between different viral strains that are that were collected. And for a given window of sampling, different sampling window, and one could evaluate distances embedded in a hyperbolic space and then monitor the radius. And it turns out that the radius is increasing as we would expect. And so one could say that the virus is also learning, and learning according to the same rules as of information theory, that it expands logarithmically with time. But then the student Anup Praturu brought me this last data point, and it's kind of lower. So where is that coming from? And then we realized that actually this is after vaccine. So vaccines were introduced here at 40 weeks. And so you can measure the impact, or quantitatively, the impact of the vaccine using measures of hyperbolic geometry by seeing how much the space has shrunk. So to summarize, what we see is that in neural circuits, viral evolution and self-development I didn't show, is that with time, the network becomes more detailed and it grows as a logarithm of time. And so in other words, many manifolds in biology have a hyperbolic axis that expands with experience. And this structure of organization facilitates, we think, communication within the network, but also the ability to transfer information to and from network. And before I would like to conclude to show you the example, just for fun, of the hyperbolic maps that were constructed. So on the left here is the hyperbolic maps of odorants, and it shows so each odorant, for example, as a set of strawberries. So a strawberry is will produce say 80 molecules, and you can measure, quantify each sample of a strawberry in terms of the volatiles that it produces, and then evaluate distances between these samples and put them on the map. And in this case, the preference, human preference, is shown by color, by purple color. So one could see that there is a continuous representations of samples that are based on odorants, and hopefully one could have expand this to other systems. So in this particular case, it was based on various fruits. Then in human perceptions, how we perceive odorants. And on the right, this is a representation of different cell types, different cells from human body. And in purple, I will highlight this is representation of different cancer cell lines. And the cells that in cancer, you might know, there are cancer cells and there are cancer stem cells. The stem cells that can regenerate and cause are more prone to cause a recurrence. So one can use these cells that are closer to the center of the space as a diagnostic for figuring out which cells are less differentiated and therefore are more prone to be these cancer stem cells. And that's the part that I talked about, neural responses. So with this, I would like to thank my group. This is Hello from Before COVID. Yuan Shengzhou did olfactory work on plant volatiles, and Huanqiu Zhang did work on neural responses in the hippocampus, and Anup Rathore developed new methods using Bayesian principles and applied it to viral evolution. So with this, I will close, and thank you for your attention and ask for your questions.
Thank you so much, Professor Sharpee. That was a wonderfully interesting talk. Many new concepts that I found very interesting. There's a question from Kathy Hunt. So she writes, I think the later commentary has explained it, but she'll ask the question. Naively, I expected the hyperbolic radius to decrease as the animal became more familiar with the environment. But now, I guess that as the animal becomes more familiar with the environment, it includes more detail in its representation of the environment. Presumably, this explains the growth of the hyperbolic radius with more exposure.
Yeah, so there is a question actually in neuroscience of how do you quantify when environment is novel and when environment is familiar. So what I think what you're astutely pointing out is that the rate of expansion slows down because it's logarithmic. So the full expression is logarithm of 1 plus t over t naught in some time. So for times t less than t naught, it expands very rapidly. You can say approximately linear with time. So it's a rapid expansion as you're introduced to a novel concept. And then for times t less than t naught, it continues to expand, but it's now logarithmic and it's slow. So your question is very relevant. It does continue to expand, but the rate of expansion changes from very rapid linear one to a slow logarithmic one.
Okay, and Kathy has typed that the added explanation makes good sense, and I appreciate it. Okay, there's a question from Roger Hilton. Is there any connection between the radius development and entropy?
Yes, so turns out, so I mentioned mutual information. So mutual information was developed by Claude Shannon during World War II and after. And it was the theory of communication, and the theory of how much you can compress representation and how to effectively transfer information over a limited channel. So it turns out that rereading this paper by Claude Shannon, his derivation of mutual information, what does it mean to convey information, is based on a tree. So one of the postulates, so what does it mean, information, and why it is so important, perhaps I should have included in the introduction, is that you can quantify with information the transmission in all kinds of things. It can be quantified during a lecture, if, like during questions and answer period. For example, if you ask me different questions, and to all questions I say yes, then my entropy is 0 and I convey 0 information. But then one can be more nuanced and start asking me different questions, and sometimes I say yes and sometimes I say no. So the entropy of my responses is potentially large. It can be 1 bit per question. But if you then start asking the same question, and I again say sometimes yes, sometimes no, then in that case, I also convey 0 information. But because my entropy of my responses to the same question, so the noise, is the same as the entropy of my responses across different questions. So to go back to your question, so the entropy of responses is roughly 2 to the n. And so the radius, we think, is directly relates to the entropy. And the difference between noise and information and entropy is reproducibility in so if each node, what is the size of the node, meaning what is the size of the variability in the position of the node as we take different repeated measurements. For example, in the case of say strawberry or neural responses, you can build a map for neurons from different segments of the data. And each time you build a map, it will be slightly different. So you can measure the fluctuation in the position of the reconstructed map. And in this way, distinguish between entropy and the information that is quantified.
Thank you very much. I actually had a question myself about the hyperbolic geometry in the brain. And is there any connection between the hyperbolic network and the actual neuron development, the way the neurons actually grow and the connectivity in the growth itself, or not?
Yes, so there are papers showing that the connections, the connectomes of different species map onto hyperbolic map. There is also, of course, a famous cell differentiation landscape for cell development, not just neurons, but it's more for a gene expression, how you start from, each cell in our body has the same genes. So as the embryo is developing, the cells become more differentiated and they form into different lineages. And then from there, they form, for example, into neurons, and from neurons to different subtypes of neurons. And so that type of process is hierarchical and hyperbolic, but also connections in the brain. There are papers that show that they're better embedded into hyperbolic space compared to Euclidean space, and they generalize better across species.
Okay. There's a question from Jan Gilman. He asks, from the plot of Fisher information per neuron, is it suggesting that the bigger and bigger large language models may not be the optimal choice for AI technology?
Well, could you please clarify why? So maybe I will provide some information and then maybe there is a follow-up question. So there is a lot of work in machine learning on hyperbolic neural networks. And so presumably that's where it's gonna go. I have some questions about the work as it currently is developed, because my understanding of how they apply hyperbolic geometry is that across layers, as you know, you have multiple layers in a deep network, but they kind of, every time they transform, they flatten it, and then they exponentiate it back to a curved space. So flatten and expand. So in some sense, I think doesn't strike me, there's something is missing there. At the same time, Professor Marr might know, I think is aware that there is this balance hypothesis in the brain where neurons have both excitatory and inhibitory connections. And when we do analysis of neural feature selectivity in the brain, we often find that each neuron is sensitive to both dimensions that increase its responses and decrease its responses. And you can model it as a quadratic model. So I'm thinking that potentially it's a reflection of the hyperbolic structure of the neural response space, because when we take these eigenvalues and then they're both positive and negative, that they cut out a piece of a hyperbolic surface.
Thank you very much. We'll take one last question. Forrest Stivers asks, you mentioned sustaining the internet with hyperbolic mapping. Does that mean you considered computer network routing protocols in your studies? If so, what routing protocol might have that been?
So I'm quoting the work from other people in that it was an inspiration for my work. And so they argue that when you do the routing, you should do the routing according to hyperbolic geometry. Now, whether, to what extent, I think that would be a private information. I don't know actually how they do it, but, that's the published work demonstrating that it is useful. And there are other series of papers that show that it improves navigation in the network, and also the response time from delivering sensory stimuli into the network. And if you think about it, the human body, the brain needs to know the status of different organs, preferably in real time, and it needs to sense whether it needs nutrients or what is the status of various organs that they need to communicate with each other. And the implication is that hyperbolic geometry would be helpful in that respect.
Great. Thank you so much, Professor Sharpee. That was a beautiful lecture, and thank you for taking time to answer all these questions so thoroughly. At this point, we will end the public part of the talk. I'll stop the recording.